And welcome everybody. I have to say it's nice to be back in person since we've been doing Zoom for a couple of years. It's good to see everybody's smiling faces again. So welcome to our course today, Life in the Extreme. We're going to explore some of the cool adaptations that various life forms have undergone or can undergo in order to thrive in extreme environments. So I hope you all enjoy the series. Our first speaker today is Dr. Jamie Foster, who comes to us all the way from the Florida Space Coast. So thank you very much for making the trek up here, Dr. Foster. We're slowly getting back the ILR in person, so we do appreciate it. Dr. Foster is a full professor in the Department of Microbiology and Cell Science at the University of Florida's College of Agriculture, although her research lab is located at the Space Life Sciences Lab in Exploration Park near the Kennedy Space Center. Her work is focused on understanding how the space environment, specifically microgravity, impacts beneficial microbe-animal interactions. Dr. Foster received her BS in zoology from the University of Massachusetts at Amherst and her master's degree in biological sciences from the University of Southern California. She completed her Ph.D. in zoology at the University of Hawaii, followed by postdoctoral fellowships in microbiology at the National Institutes of Health and NASA Ames Research Center. Dr. Foster has conducted 3 spaceflight experiments aboard the Space Shuttle and International Space Station, which examined the effects of microgravity on the symbiotic interactions between microbes and their eukaryotic hosts. And her talk to us today is the effect of microgravity on animal-microbe symbiosis. Welcome, Dr. Foster.
Well, thank you very much, everybody. And I hope you liked. I scheduled a launch about an hour ago for you guys to watch. I hope you all enjoyed that. Elon and I get along really well, so I scheduled that for you. But thank you very much for having me and being part of this session on life in the extreme. Do we have the slides up? Awesome. So you might think, what is a little squid? How extreme is squid? But what we're really trying to do is understand how the stress of living, working, and being in the space environment affects our normal healthy interactions with microbes. And so the model, I started out, as we mentioned, she gave a wonderful introduction. Thank you so much. And I started working on this for my PhD research, understanding how microbes talk to and communicate with animal tissues using this squid as a model. And when I was a graduate student, I thought, this would make a great model for space biology research. So 10 years later, I was finally able to actually start that research here at the University of Florida. So I've been very lucky to be part of a small group that's located off campus down next to the Kennedy Space Center. So one of the things that makes this all very extreme is when you go into space, your orientation, your body fluids, your, where your blood's going, everything is messed up. And that's the true for you, the astronaut, or the microbes that are inhabiting and interfacing with your body. So living and working in space, as we slowly turn our attention away from low Earth orbit, and now we're looking at the moon with the upcoming Artemis missions, and very soon the Mars missions, we're really trying to understand the effects of the space hazards, microgravity, radiation, the psychology of space, all of that on people and the spacecraft with which they inhabit. And so there are a lot of questions we have to tackle as space scientists to keep and help people stay healthy in space. And I really approach it from a microbiology angle in what microbes are people being exposed to. Crew come and go on the spacecraft, and they will. This is a mock of the new space station that's going to be orbiting the moon. It's called the Gateway. Construction on that is already happening. So what are the microbes that you're going to interface with other crew members and the spacecraft? What factors are going to control the abundance of those microbes, the persistence and the diversity of those microbes? And how are they going to affect human health? 99% of all the microbes in our body are beneficial. What happens when you stress them? Do they become more pathogenic? Do they start degrading building materials? Do they form biofilms? And we have to worry about how our life support systems are going to function. So all of these are big questions that are beyond me, just one person, but that NASA, the University of Florida, and other researchers are focusing on trying to answer. But for me, I'm really interested in how microgravity, specifically that space hazard, that space stressor, is affecting beneficial interactions between microbes and their animal hosts. And so, as I just mentioned, most of the studies that have been done in the space environment have focused on pathogens, Salmonella, E. coli, Pseudomonas, but they're only representing 1% of all the bacteria that interface with your bodies. So what's happening to the other 99% of the taxa that are interacting with the cells of human tissues and also plant tissues? I'm focusing on animals today, but there's a huge line of research here also looking at the microbes that interface with plants as well in space. But my question is do mutualistic bacteria, are they altering their physiology? Are they showing any signs of pathogenesis when they're stressed in microgravity? Because a lot of the signaling molecules that are used by both pathogens and mutualistic bacteria have a commonality. They use a common language. These things are things called lipopolysaccharides or peptidoglycan derivatives. We call them MAMPs, or microbe-associated molecular patterns. So the language of beneficial microbes and the language of pathogens are often the same. But where is the balance between health and disease, and does one stress push you over the edge towards pathogenesis? And those are the kinds of questions. And so how does that balance happen? And also, by taking gravity away from, gravity has been the one constant on our planet for 4.6 billion years. Gravity's always been there. It's always been constant. It's a driver of evolution on this planet. So when you remove that, is it obscuring something that we wouldn't normally otherwise see how microbes are talking to? And so by kind of using microgravity as a bioassay, we can start to see how that low shear, low turbulent environments of microgravity might be affecting the interactions between microbes and animals. So the lessons we learn in space, we want to bring that back to Earth and help us understand problems here as well. So it's not just about keeping astronauts alive. It's also about using those lessons learned to have new technologies, new tools, new pharmaceuticals for us here on Earth. So what you might think, okay, I can understand microgravity affecting a human physiology, but a microbe, it's so small. How is it could possibly be affecting the physiology of a microbe? Well, a lot of that work was done on Salmonella that started it, E. coli, Pseudomonas, a lot of typical pathogens. But a trend is emerging of all the microbes that have been tested. And what we find is that when microbes are grown in liquid cultures in modeled microgravity or actual gravity, their growth rate increases. They grow faster under all gravity conditions, microgravity conditions that we've tested. They have a reduced lag phase. That's the transition between their growth phases, and they increase exponential growth. It also, as I mentioned, it creates a low shear environment, and that seems to have an effect of it. They become more resistant to antibiotics. They seem to be much more resistant to environmental stress, like oxidative stress. And they also, this is in particular E. coli, seems to survive longer in host macrophages. And macrophages are immune cells that often, through their innate immunity, take up microbes and degrade them. But unfortunately, a lot of microbes, when they're in space or in modeled microgravity, survive longer in these immune cells. And of course, they have differential gene expression. And unfortunately, some microbes show increased signs of virulence when they're flown in space. So this was just a graph. I'm not going to go too deep into data here for you, but this was some data from a researcher named Cheryl Nickerson on Salmonella. And I apologize, this is a little faint, but she took Salmonella that was flown up into on the space station and brought them back down and exposed them to mice. And in all cases, the LD50, or the rate at which the microbes were killing the mice increased, and they had a much higher infection of microbes under the under the microgravity conditions. And basically, the salmonella increased their virulence and became more pathogenic. So that's a problem. You have microbes potentially getting more pathogenic in the space environment, and you also have astronauts' immune systems becoming slightly compromised. Their macrophages aren't working so well. So you have to, so in order to keep people healthy, you might have this double whammy here of microbes becoming more virulent and the immune system not functioning as well. So that's why it's really important for us to understand these processes of how microgravity is affecting microbes. But I also really want to not just focus on these pathogens, but also on these beneficial microbes. So to do this, I have used the model between the bobtail squid. This is what an adult looks like. The first picture you saw was a juvenile, one that was about a month old, but this is an adult squid. And the squid actually have two symbiotic organs inside them. They have what's called the light organ, and then they also have, the females have what's called the accessory nidamental gland. I won't talk about this one. There's some research at the University of Connecticut that's starting to look at this symbiosis, but this is the one that I have been focusing on for my space research. But this light organ has, it almost looks like an eye. It has a central core of where the bacteria live. They have a reflector, and the bacteria are actually luminescent. And I'll show you some pictures of that. They glow in the dark. And the squid uses the light. It shines the light downward through a lens-like material. And what the squid does is it shines the light downward into the water column. So any predator that's kind of looking up at the squid sees nothing. The squid is basically kind of using it as like a Klingon cloaking device and kind of hiding from the predators by matching the downwelling moonlight that's coming. So basically it's like shining a flashlight in a big light and you can't quite see where the squid is. So this has been a kind of a defense behavior for the squid. But what's interesting is it's just a single species of bacterium. Unlike our guts, unlike most symbiosis, where there's thousands of different taxa, it's just one symbiont colonizing this light organ here. So it makes it easiest. It's a very simple binary system where you have one host, one bacterium. So you can add the partners together, you can use antibiotics and cure the partners, take them apart. So you can really tease apart what this one bacterium is doing to communicate with its host. And we can also look at it's also simplified in that squid don't have adaptive immune systems. They have what's called innate immune systems. So it's a simplified immune system for us to start to track how the microbes are responding, how the squid is responding to the presence of microbes. So that's why we've been using this as a model for 30 years to study symbiosis. But as I mentioned as a grad student, I thought this might be a great model to start exploring how stress is affecting that. And here's some just kind of prettier pictures of how the microbes, so we've labeled this with a protein called GFP, green fluorescent protein, and they start to aggregate within hours. So you have a baby squid, it's hatched, and it's exposed. And one thing I'm sorry I forgot to mention is that on the surface, when the baby squid are born, they have these fields of epithelial cells called ciliated epithelial cells. We're really original in how we name things. But they kind of extend outward from this light organ here, and they're constantly bringing bacteria into the vicinity of pores on the surface of the light organ. So these cilia are constantly entraining bacteria from the environment to come close to the light organ. And Vibrio fischeri, Vibrio fischeri gathers and aggregates outside these pores, and they kind of form these little cha-cha lines, and they just kind of go into the light organ and colonize. And by 12 hours, the squid has become colonized, and it becomes luminescent. It has the ability to glow in the dark. So we wanted to use these features to see how the stress of microgravity, so here are those ciliated structures used for recruitment. Once the animal gets colonized, and we call them aposymbiotic when they don't have the right microbe. And if you never give them the right microbe, if you put them in filtered water or whatever, they never undergo any kind of developmental changes or anything. But when you give them the right bacteria, when you give them Vibrio fischeri, they kind of lose these structures and kind of get this bowling ball effect here. But this is the model system I thought would make a really good model for studying the stress of spaceflight because of this binary symbiosis, this one host, one symbiont. And here's just what the animal looks like as a juvenile. So how do we simulate microgravity? As you, Paula, mentioned, I have had the opportunity to fly a few space flights, but 3 in a career of, 3 space flights in the last 17 years, that's not a great, way to do all my science, right? So we have to do more experiments. And how we do that is we use what's called modeled microgravity or simulated microgravity. NASA a few decades ago came up with these, they're called HARVs, high aspect ratio vessels. And they're little bioreactors here. You can see one spinning, and we can either put them in vertical position or in a horizontal position. So what's happening is as it's rotating like this, gravity is pushing down, but the fluid forces, there's seawater or media in here, pushing back up. So the cells or the squid or whatever's inside there gets suspended. It's like they're jumping out of a plane and they're in free fall constantly. So that simulates the weightlessness effect on the cells or the microbes or the squid, because we can put, as you can see here, these are actually baby squid. They're very tiny when they're born. That's another good advantage of them for space flight is they're only 2 millimeters in size when they're born. So we can put hundreds up at a time in a little box this big. So we can simulate this free fall microgravity, and that allows us to do lots of replicate experiments, hone down our hypotheses, and kind of come up with some foundational research that then we can fly. And here's the box. This box is about that size. And we put the squid in little, it's like bag of squid. And we put them in these little bags, aquarium bags, and we have pumps here, and the whole experiment is automated. And I'm going to talk about that at the very end, because we just got the data back, and I don't have everything figured out yet on that, but I can share with you what the experiment was about. So anyway, we use these these HARVs to replicate the conditions of microgravity, and then just to make sure we don't have any bioreactor effects, we then turn them so they're still spinning, but they're getting the full force of gravity on them. So though, whenever you see a gravity control in any of the data I'm going to show you or pictures I show you, that's what we're talking about. So what we found when we started exposing the colonization process of this bacterium and squid to this microgravity is that some things were sped up and some things slowed down. One of the first things we looked at was the immune system response. We know astronauts have delayed immune systems in space. What was happening in the modeled microgravity? And sure enough, I'll just go back here. The hemocytes, the immune cells of the squid kind of infiltrate the light organ. But in in normal gravity conditions, they do it fast. Within an hour of it being exposed to bacteria, the cells are moving where they're supposed to go, and they're tracking to the tissues of the squid. But it's delayed in modeled microgravity. We're having a 10-hour delay. The whole thing should have been colonized by 12 hours, and yet here we are having a delay. So the immune system of the squid is also delayed, just like the immune system of astronauts. So that gave us a sense that maybe this might be a good model system for us to explore. And then the other thing that happens is normally the bacteria trigger the cell death through apoptosis of those tissues used to recruit the bacteria. Well, that happens faster. Normally in microgravity, you start to see some cells dying around 6 hours, and then that peaks around 16 hours. All the cell death is happening within hours in the stress of microgravity. So that part of so we always thought that the hemocytes might be bringing in the signals to cause the apoptosis and the loss of cells, but it turns out it's not. It's uncoupled. Something else must be causing the triggering of that apoptotic cell death event. So here's a case where treating it as a bioassay of stress, we can start to tease apart things we always thought were connected, that they're actually uncoupled events. And then the bowling ball effect happens a little bit sooner too. Those arms die through apoptosis, they're regress. So we had some phenotypes that we could start to play around with, that stress of microgravity was causing changes in the normal developmental timeline of the animals. And the other thing, so then we started, and I don't have a lot of time, so I'm just going to talk about apoptosis, the cell death event that's happening. And normally, again, you have these structures here that are covered in cilia here. And when the bacteria are added, they start to die off through a process of apoptosis. But what we found was we could simulate that cell death event using just a single molecule of the bacterium called lipopolysaccharide. It's one of those MAMPs that are often used in pathogenic cells to cause cell death. So was microgravity changing the production of this bacterial molecule? And are there changes in the response of the animal to that molecule? And what we found, and so I'm going to tell you a little story about the effects of spaceflight on the production on the bacteria molecules, and then I'm going to tell you another little side story about how the animals responded. So that's what we're going to talk about. So when bacteria are swimming through microgravity, they are whipping around their flagella like crazy. And through that process, they're releasing these little vesicles. And you might have heard of these things called outer membrane vesicles. They're being used or considered for a lot of drug delivery, vaccine development, because they're just these little bits of cell membrane that you can load up with different contents. So maybe you can put a drug in there and deliver it. So a lot of people are now focusing on outer membrane vesicles, how they're made, how you can produce them. And it turns out under modeled microgravity, you can produce lots and lots of them. They actually increase their production of these outer membrane vesicles under the stress of microgravity. So we were very curious about that. And what we found, here's just a test. So you can, in those outer membrane vesicles, like I said, they're bits of the membrane. So they contain this lipopolysaccharide that we know affects the animal. And it turns out that the red here is the modeled microgravity, that in all cases, all timelines that we tested, the microgravity caused more of this LPS, more of the OMVs to be produced. So that stress was causing and you can kind of see, here's just a picture, some pictures of the Vibrio fischeri, and they've just got all these vesiculation, these hypervesiculation, these little droplets kind of budding off of them. So that was something that we found was a big change, is this increased production of endotoxin. Lipopolysaccharide is an endotoxin, and these outer membrane vesicles under modeled microgravity. And then what we did is we wanted to see, so we knew it was tied to them swimming around. What happens if we stop them swimming? So we made mutants in the bacterium that basically targeted their flagella. So they either couldn't make a flagella, this is called a FLA mutant, or they couldn't turn the flagella through a MOT-B. So then we said, okay, if we take away their inability to swim, can they still make all this excess endotoxin and OMVs? And it turns out that we did. This is the wild type, which is the parent strain or the natural strain of Vibrio fischeri, and we knocked out that ability to make or turn their flagella, and we did knock down quite a bit of their ability to make this increased amount of endotoxin under the stress of microgravity, and also OMVs. But we looked a little closer. This graph here is this one blown up a little bit. And what we found is we still see more in modeled microgravity than gravity control. So that suggested that our major hypothesis that flagella were the reason why these bacteria were making these little endotoxic vesicles, but there might be other ways that these products could be being made. So we started to explore the health of the bacterial membrane under the stress of microgravity. And so here we have just untreated. So again, we have our ES114s. The ES stands for Euprimna scolipes. That's the Latin name of the squid. And 114 was just the 114th strain we isolated from the squid. So this was our wild type, the same here. And then our mutants here. And under normal conditions, gravity conditions, when you don't disturb them, there's really not a lot of difference between the gravity and the modeled microgravity. However, when you stress, we use chemicals like polymyxin or also Triton X, when you stress the membrane, the cells growing in microgravity burst faster. They basically, here's a viability stain, so here's gravity here, and then the cells exposed to the microgravity, which is the dark ray, are killed they're much more susceptible to the disruptions to their membrane. So something about being in the microgravity environment is causing them to shed a lot of their membrane, making their membrane weaker and more susceptible to different agents. So again, we're seeing some physiological changes that maybe we can take advantage of as we try to keep, as we start to explore potential applications for some of this research beyond just spaceflight. That was just to show that. So that's the little story I wanted to say, is that we are now exploring the mechanism. So now we're looking at the membranes, and we've created a whole series of mutants that are looking at different defective parts of the membrane. We're looking at lipoproteins and outer membrane proteins. We're trying to suss out what the second mechanism is of why the cells are so much more susceptible to bursting and lysing in the modeled microgravity environment. And hopefully someday we can do this experiment in space, but that's going to be a little tricky. So then the second part of what I wanted to tell you is what happens to the host on this? Okay, so the physiology of the bacterium, they're producing more LPS, their membranes are getting a little messed up, they're producing all these OMVs. How is that affecting the host, and what's changing in the host side of things? So as I mentioned, the bacteria are triggering a developmental process called apoptosis. This happens in our bodies all the time. Cells are undergoing programmed cell death, where if a cell is damaged beyond repair, it is controlled, it is destroyed in a very controlled way and removed from the body so that it doesn't cause inflammation or other problems. And in animals, there are two ways. There's what's called an intrinsic process, where some signal from the outside of the body is funneled in, and there's intrinsic, where some sort of internal stress is causing the cell to die from the inside out, as opposed from the outside in. So our hypothesis was we expected a lot of genes associated with extrinsic apoptosis, and that lipopolysaccharide that's being increasingly shed from the bacterium might be triggering that process. So we started, one of my graduate students, we just got the genome sequenced of the squid just a couple years ago. We published that. Cephalopods have very weird genomes. They're huge genomes. They're bigger than ours. And so we started to explore the genome for potential candidates to go after. And we found tons of candidates that I'm not going to bore you with all these candidates. But we found this one group of enzymes called caspases that are absolutely critical for this apoptosis process. And we wanted to explore how they were being affected by this microgravity environment. So the role of these caspases, are they part of the process here of why the bacteria are triggering this accelerated apoptosis event in the modeled microgravity condition? So we approached this in a few different ways. One is we looked at the expression of all those genes in the squid under the different gravity conditions, so under gravity controls, as well as this stands for low sheared modeled microgravity here. And so we used a technique called nanostring, where you can target as many genes as you want all at once. You're not sequencing everything like an RNA-seq, but you're sequencing a targeted amount. It's a little smaller way to go after specific genes. And then we also looked at the proteins, the enzymes actually being, and whether they were active and whether their activity changed. But the results of so we looked at the expression of these caspases, and we found that in the model's microgravity, green means they're highly expressed, red means they're very low expressed. There is some increased expression in the gravity controls, but they are much higher expressed. So that something about the stress of microgravity was causing an increased expression of these enzymes. And these are the enzymes that degrade your cells during apoptosis. They come in at different stages of the process, but they are what we call executioner enzymes that actually kill the cell. And they are being up in modeled microgravity. So is that a concern that some of the cells are actually undergoing, this increased expression of these executioner proteins? That was something we wanted to follow and explore more. But just because you have a gene and the gene is turned on doesn't mean the protein is active and they're actively degrading your cells. So we wanted to explore the actual activity of the protein itself. And so to do that, we use a series of kits called glow kits. They're just something you can buy commercially. And they go after the different types of these caspases, and we can try to suss out which caspase is doing what and when and where. And so in all cases, we took animals that had just been hatched, animals that had never been exposed to bacteria, and animals that were exposed to bacteria, and measured the levels. And we were able to see in all cases that these enzymes go way up when bacteria are present. So then what happens when you stick them in modeled microgravity? Well, they too, their enzymes, their productivity of, not only do they have the transcript, but their enzyme activity also increases under the modeled microgravity controls. So something about microgravity is causing those cells to die faster, and that's happening through a process using these different caspase enzymes. And so we just published this, but this has helped us come up with a pathway of how the microgravity is altering the physiology of the squid cells, the epithelial cells that are interacting with these bacteria, to try to suss out why microgravity is causing this accelerated cell death. But we also wanted to see, can we stop this process? Can we inhibit this microgravity effect from happening? And so we used a series of different enzyme or different inhibitors, different peptides that could block the function of this enzyme, and then repeat the experiment to see if we could kind of control, can we mitigate the effects of microgravity on the animals? And the answer was, to some extent, we could. We tried a bunch of these different caspase inhibitors. Here are our controls, and we did knock down. So here on this axis here is all the dying cells. We just counted all the number of dying cells, and we used a stain and some microscopy to visualize them. And we were able to lower the amount of cell death using these inhibitors, but we weren't able to completely block it. So that suggests there's something else working as well, and that we think is a molecule called capsaicin that we were also exploring. But we were able to lower the amount of apoptosis when we added these inhibitors. And I know this is a little messy, but again, we seem to have knocked down the ability of microgravity to induce the amount of this accelerated apoptosis. So we were able to mitigate this spaceflight response, or this microgravity response in the animal in response. So we know we have a lot more work to do because we didn't knock it out completely. There's still dying cells happening here, but it's our first step towards trying to control. And actually, the paper has already come out. It says in revision, but the paper just came out a couple of weeks ago. So we are working to take steps to mitigate the effects of microgravity on these cells. Okay, so I gave you, that's it for the data. Data's gone. So now we can talk fun things, right? So one of the things we found was that, just to recap what I just told you, and now in the last couple minutes of the talk, I'm going to think forward on how we're going to take this research and research like it and think about how to apply it to research on the moon. But just to recap, we are seeing that modeled microgravity is increasing the amount of production of these bacterial signal molecules that can cause problems for the host tissues. In particular, MAMPs like lipopolysaccharide. We're going to explore other molecules like peptoglycan derivatives and other things, but right now we're trying to learn a little bit more about that process. And we know that these MAMPs are packaged in these outer membrane vesicles, and they're constantly being shed in modeled microgravity. So we're looking at ways, can we mitigate that? Can we use that? Is there some sort of applied technology that we can use microgravity to increase our production of these outer membrane vesicles? And maybe there's bio applications for that as well. And also we know that microgravity is weakening the membranes of the bacteria. And we think that the membranes are weakening because the cells are stressed and they're trying to get rid of waste products. And we know that there's a phenotype on the host side. They're producing gobs and gobs of this enzyme called caspase, which is damaging the cells and causing apoptosis. And we know that it's not just the genes, but there's also activity as well. And so we are looking at trying to inhibit those activities using various pharmacological agents. So that's what I've been working on. And after we kind of I go through these slides, I'll tell you briefly about the space flight. I can't talk about too much just because I don't want to steal my students' thunder because we're working on getting it published. But one of the things I'm also involved in is a group called BLEO. BLEO is Beyond Low Earth Orbit. For the past, obviously, for 20 years, humans have been orbiting the planet. You know, they've been in low Earth orbit. We have the International Space Station that's retiring in a couple years. And we have 4 new commercial space stations that are coming online. The Chinese have their space station. And so there's going to be a lot of commercial space applications. People are going to keep doing their science. But NASA is kind of handing the keys over to low Earth orbit to these commercial entities like Northrop Grumman's, NanoRacks, Blue Origin, which is Jeff Bezos, and of course SpaceX, which we all saw go launch today. And so what happens, so NASA's kind of saying, okay, you guys do low Earth orbit, we're going to focus on the moon. So how do you deal and do these kinds of experiments on the moon? So I'm a part of a working group that is thinking about the technological challenges to doing research outside our low Earth orbit, where there's going to be less crew time, harder, it's going to be fewer missions out there, and you're probably not going to get any of your samples back. So how do you do science when you have all these limitations? And so, but there are people working on this, and there's some really cool experiments that are happening now. One of I posed several questions to you at the very beginning about how do we track microbes, what's happening to them. And I just told you a story that microbes' physiology is changing in microgravity. So we have to be conscious of that. We have to be tracking them. And so there's a program called the Microbial Tracking Program. There's actually a third one now. This is just a slide for the second one, where and the International Space Station inside, there's all these little targets all around, and they swab those targets throughout the space station, and then they package them up, send them back, and the microbiology of the space station is constantly being observed. So as crew come in and out, the microbes are changed in response to the crew, because the crew are primarily inoculating the space station. So anyway, this is a program, and how do you take a program like this, where literally it's astronauts, wiping down the walls and sending it back? How do you automate that? And actually, here's just a picture of some of what it looks like, some of the microbes growing on the walls, and they have a very complex, pattern of monitoring and stuff, and they're finding billions of microbes all over the place, and they're tracking whether or not they are pathogenic or not pathogenic. And so there's a lot, and here's just some of the dominant taxa that they're finding. But it is very important to NASA to keep track of what's growing inside the space station. And they're also tracking the plants. Are now more and more food crops are coming up. They got lettuce, tomato, radishes, all these things that are growing. So that's also depositing microbes. So that is something that they're very interested in. So how do you do that on something like the moon? Well, they're working on fully automating this. This is called the Voltrax. This is in partnership with a company called Oxford Nanopore. And so they are using artificial intelligence to control this process. So all the astronaut crew right now, this is a little robot that floats around the space station right now, but they want to be able to use, maybe a little probe would stick out, they could swipe the wall, and then they can insert it in this. This is called the Voltrax. It can actually do the DNA extraction, the isolation, and the sequencing all in one little box. And then the data could be electronically transferred to or sent to Earth. So this is something that they are working very hard to create a viable microbiology monitoring program on the moon, or on all the spacecraft that operate on the moon. So that's something that is coming. But it becomes an interesting question. This is more of a, I'll kind of leave with this is that will working and living in the moon require microbiome treatments? Will we have to intentionally engineer specific microbes to prevent, biofilms from forming in the water systems? And so can we use what we learn from the microbiology, little things like what I'm doing and what others are doing, to help shape and direct and almost a spacecraft probiotic to help control a lot of the microbial populations? So this is something that they're thinking about. What microbes could this be? And how can we synthetically engineer, microbes to do the work for us and to control? Can they make probiotics if you're halfway to the moon or you're stuck on the moon? Can you, your vitamins are going to degrade over time, your probiotics. So can you engineer or create new products on the fly in the moon? And there's a lot of money going into to investigate those kinds of questions. So instead of maybe you don't think of it as a microbiology problem, maybe you think of it as a biodiversity problem, and you control what species you have and forget about how many species, or how many cells you have up there. So there's a lot of questions and a lot of people thinking about that. But there's going to be a lot of platforms to do research. So there's going to be a lot of free flyers. Those are single shot satellites that all the data is just beamed back to Earth. There's the Orion, which hopefully will launch soon. It hasn't still sitting in the vertical at NASA on the launch pad there. And then Gateway is the new space station that's going to be built around the moon. That's coming. Parts of it are already being built. And then, of course, with once we get to Artemis 4, that's supposed to have people landing on the moon. We'll have landers and hopefully maybe a lab. So there's a lot of this is happening right now. I mean, we're here on this chart right now. Or well, I guess we were here because it hasn't launched. It exists, but it hasn't launched. And so there's going to be a lot of great science. And there's also all these commercial companies that have built little rovers and platforms for you to do your science and your experiment. So all the experiments that I just talked about with the little squid and the bacterium, maybe we use tardigrades, maybe we don't use squid, but there's lots of ways in which you can get, scientists are going to be able to get their science done in this platform. And that's all I was going to tell you. And I thank you very much for your attention. So all this work was funded by NASA, and Redwire was the company that helped us build our actual hardware here for launching in space. And then my student, Maddie Vroom, who did all that LPS work and the membrane, she was supported by the Florida Space Grant Consortium and the members of my lab. So thank you for your time. And if there's any questions, I'm very happy to address them. Yes, ma'am.
Thank you.
Yes.
Oh, yep. I love the last name of your student, Vroom.
Yeah, Vroom Vroom.
Full speed ahead.
And
there.
she was always working at warp factor 9.
Very interesting. So how does the baby squid distinguish and recruit only one species of bacteria into its little cell when it's bringing in seawater, which must have billions of bacteria?
No, that is something that we've been working on, and we don't have all the answers on that. But what happens is this, so in that little aggregate, that ball of cells before they form that little cha-cha line and colonize, that is an extremely oxidative stress. It's a nasty place to be if you're a microbe. They're producing lots and lots of nitric oxide and oxygen radicals. And it turns out Vibrio fischeri has pretty good catalases that can detoxify that. So that is one way where a lot of the microbes can't survive in that little. So the bacteria then also produce the Vibrio fischeri produces a biofilm that allows them to kind of hunker down in that little aggregate environment. And then so a few break away and then colonize. So it's a very oxidatively stressful environment. And then there's also, once they're traveling down through the tunnels to get them into the light organ, that also is constantly being infiltrated with reactive oxygen species. So the squid kind of creates a hostile environment to anything that's non-competent, that doesn't have the ability to deal with that. That's just one of the few ways.
So so there's macrophages that are all part of this, right? So
What are they doing?
well, so they're kind of sampling what's coming in, right?
Yeah.
So if they suck in a Vibrio fisheri, they're going to say, okay, well, we want that, right?
Right. So you're absolutely right. The hemocytes are flooding the whole system, and they sample some of the Vibrio fisheri. And we don't have the full understanding of that conversation that's happening in there, but once they learn what a Vibrio fisheri is, when the animals are an adult, the adult macrophages or hemocytes never take in Vibrio fisheri. So the other thing I didn't mention is that once colonization actually only happens once when the animal is born, and it retains, so every morning, like a tube of toothpaste, it spits out the light organ. They collapse those muscles, and like a tube of toothpaste, they spit out 95% of all the microbes in their light organ to the environment. But it keeps 5%, and that 5% regrows up. So you only have to kind of keep your defenses up a little bit in the beginning. And then we think that spitting out of 95% helps repopulate the environment, and also you don't have to feed them. So during the day, so at night you're using the light to hide from predators, but during the day you don't need to feed your microbes. So you can keep that 5%, and they grow up over the course of an evening or the day. So the squid are nocturnal, they're out at night, and then they bury in the sand during the day. So we think it's a two-way process of not having to feed them. And but that we know that the macrophages, we think, are curating it, but that's only in the beginning. And then once the symbiosis establishes, there's no recolonization that happens. There's a lot that happens. It's a simple symbiosis, but there's a lot going on. Yes, sir.
So I have lots of technical questions. The harves, that's here that you're
Yep, I have them in my lab. Yep.
Okay.
There's quite a few labs that have them here.
Yeah. All right. So I guess the question is, how good a representation is that of what you're really trying
Absolutely
to look for? And I mean, in the actual experiment, presumably your organisms, you send them up in a mouse?
In a mouse? No.
You don't. Okay, so I guess that's what I'm asking.
It's all done with the squid. We sent the squid.
Right. Yeah. Okay. I'm sorry.
So the squid or the mice?
Let's do it. Let's do it in the squid. It doesn't make any difference in this matter. So you're looking at its pathogenicity in situ, essentially, in space.
In vivo, actually.
Yeah. Well, whatever it is.
Oh, yeah, yeah.
Yeah. So when you said that in the space vehicles, they have this wonderful collection of microbes.
We're having trouble hearing Ken.
Are those all from the environment? They're not introduced except adventitiously.
I see what you're saying. I think we keep the so when I fly my experiment, it's triple containment because we try to prevent the back. So actually, I can even show you. Is it possible to get the slides back up? Sorry, I can show you a picture of what it looks like. So the first question is are the harves really an ideal model? Do they fully replicate the conditions of spaceflight? And the answer is no. So we have several platforms in addition to harves. There's what's called a random positioning. There's different kinds of clinostats that are randomizing the gravity vector to try to disorient the animals and try to mimic these kinds of environments. But actually so I'm going to go one more. Oh, maybe I can't. So actually, can we do one more? Okay, it should work. This is the little bag here. So here is a baby squid here. There's my hand here. And so we put these in those containers, and then they're in a third container, and then they're in an actual even bigger container here. This is Megan, the astronaut who actually did our experiment. So there is triple containment to try to prevent any a contamination from outside in and inside out. So we really don't want that question of contamination. So we have, we've really tried to, and this is all sterilized beforehand and so forth. So we work really hard to, now she is not in a sterilized, so she is the one that is bringing up the microbes, contaminating the walls and stuff, and that's just the price of human exploration. So that's something that we do think about contamination, and we acknowledge that the HARVs are not a perfect model, but it allows us to down sample our hypotheses so that when we do the space flight, we have a general idea of what might happen. I think there was a gentleman over here who had a question. Oh, no.
Okay.
Oh, sorry.
It's on already? Can you guys hear me? Okay. Let me get this. You alluded earlier to the bacteria reproducing, and I was wondering if that confounds your experiment because of the weakening of the membranes. At the same time they're reproducing, and you're trying to figure out whether they're
Shedding all this material.
Yeah, shedding all this material that could also be shed while they're reproducing, right? So do they reproduce? What is the turnover time?
Yeah, so sorry, I don't mean to be so loud. They increase their graph. So normally they go in exponential. It takes them about 16 hours to get into stationary phase. And they just reach a higher density, and they stay in longer in exponential phase. But when we did those experiments, we normalized to cell number to make sure that it wasn't. So you're absolutely right. We did normalize the cell number. So we knew we were starting with the same number of cells, and we were getting the increase in these vesicles. But you're right. They grow pretty fast. So if we didn't normalize, we would definitely probably it'd just be because there's more cells and more division. Oh, thank you.
Yeah, Pushpa, go ahead.
Yes, ma'am.
Yeah, in another course, parallel course to this, we started on the microbiome, human microbiome, yesterday, which is also a new field for most of us.
Yeah.
And I was wondering if any studies are being done with the human microbiome, the astronauts going up, would that be affected? Beneficial, harmful, what?
Are they doing the fecal
Any
samples
studies?
when they?
And also others. Yes, they are doing human microbiome research. And unfortunately, the numbers are, so NASA's super protective of the astronauts and their privacy. And so you have to kind of get an astronaut to agree to be swabbed everywhere. And so it's not just fecal, it's also nasal swabs and skin swabs and things like that. So there's probably, and also there's more male astronauts than female astronauts. So I think they have a statistical problem, but they are definitely trying to do human microbiome research. And actually there was a fantastic study published on Mike Kelly and Scott Kelly. They were twins that flew up, and Scott Kelly spent a year up there. And so they went to the microbiome, and they went to his genome and saw all sorts of differences. So I think you're going to see more of that. And people are also looking, it seems the microbiome can affect osteoporosis and all sorts of other conditions. So Parkinson's, neurological, diabetes. So they are, and also we haven't even talked about viruses. So this is a huge field.
So are there any preliminary results from the astronauts?
There are a few biosignatures. For example, there was a study that just came out that suggested they were looking at osteoporosis. And they were they took data from Earth in osteoporosis. Unfortunately, it was primarily females, and astronauts are primarily male. But what they found was a few bacterial signatures that only in osteoporosis cases do you see this one bacterium. So we don't know if the bacterium is just a canary in the mine or if it's causing this. So this is where we're at. We're just seeing signatures or correlations between types of microbes and the conditions, but we have no idea about cause and effect.
Well, yeah, we are just finding out most of the effects of microbiomes on Earth in humans. So it's very fascinating.
It's really funny because 30 years ago, I was presenting my little squid. I was a graduate student, you know, when I would go to a microbiology meeting, everybody would come up to me and say, wow, that's great. You know, that's really cool stuff. And then I'd go to an animal developmental biology meeting and no one wanted to have anything to do with me. Microbes controlling the animal development, forget it. They didn't want to have anything. So I think we've come a long way in 30 years.
So I guess one thing I was curious about, I think you mentioned early on in your talk, some of the earlier work where they were looking at E. coli, salmonella, you know, taking things up in space and bringing them back.
Yeah.
And my problem with that was always, how did they preserve, you know, what the changes were from microgravity by the time they got back down to Earth? I mean, if you're preparing like RNA, that's one thing, you can do that up there and bring
Yeah.
it down. But like whole organisms that are live.
Yeah, it's not perfect. So they would do the experiments in my building, actually. So I am literally like a mile from where they land. This was all on space shuttles, so they would literally land the space shuttle, take the samples, and drive them over, and then infect the mice in my building. So we had an animal care facility. So these were a lot of the experiments were done on, but they were, you know, they had hypergravity of landing, they had the stress of this. So you're absolutely right, it's not perfect. And I guess when they did outgrowths, that behavior was lost. So that increased virulence didn't persist in the cultures. So it was definitely, and since then they've looked at, like, I think they've done this on tissue cultures together up there. But rodent experiments do exist, and they do fly rodents up there, and they have special cages up there. But yeah, you have that caveat of potentially losing that phenotype.
Yeah, and then to address Ken's question too about how well you're approximating microgravity with your devices. So you had a couple of experiments that did go up in space. So were you able to correlate, you know, from those 3 experiments, any of what you've seen with your artificial?
We have. Okay, why not? I'll tell you the big secret. Should I tell you the secret? No, don't be nice on Zoom out there. Don't report this. No. No, what we found on the ground using our models, microgravity,